Welcome and thank you for joining us today. My name is Rohit Mehra, and I'm honored to open this GUPS Distinguished Lecture Series event session organized by our GUPS Education Committee. At GUPS, our mission is rooted in a commitment to improving patient care through education, research, and promotion of best practices in urologic pathology. We are proud to serve as a hub for collaboration and innovation among professionals dedicated to genitourinary diseases. Today's event is part of our new Distinguished Lecture Series, a cornerstone of GUPS programming. Through Distinguished Lecture Series, we learn from visionary leaders in the field like translational scientists, clinicians, and pathologists whose groundbreaking work continues to shape our understanding of GU pathology. And these leaders are not only prolific contributors to literature, but also active voices in shaping the future of cancer research, clinical care, and pathology. Their insights have indeed redefined how we approach tumor biology and diagnostics. Uh, today's, today's session addresses a timely and complex topic. What can we learn about prostate cancer biology and etiology from population based or patho-epidemiology studies? This session is being moderated by my outstanding GU colleague and GUPS Education Committee member, Dr. Tamara Lotan from Johns Hopkins University. I'm now requesting Dr. Lotan to please welcome and introduce our distinguished speaker, Dr. Lorelei Mucci, who has kindly agreed to bring her expertise and discuss this topic with us. Over to you, Tamara.
Great. Thanks so much, Rhett. So it's really a pleasure today to introduce a colleague and longtime collaborator, Dr. Lorelei Mucci, who's a professor of epidemiology at the Harvard Chan School of Public Health. At the School of Public Health, she serves as director of cancer epidemiology and cancer prevention program, associate director of population science at the Dana-Farber Harvard Cancer Center, and also director of strategic research partnerships at the American Cancer Society. Dr. Mucci received her doctoral degree in epidemiology from the Karolinska Institute in Sweden, where she gained expertise using nationwide health registries to examine cancer etiology. And formed a long-term partnership with epidemiology colleagues in the Nordic countries. For the past 19 years, she's really focused on using integrative molecular epidemiology approaches within cohorts in the United States and also globally to investigate research questions focused around cancer etiology, mortality, and survivorship, with a particular focus on prostate cancer. I think she has a wonderful perspective, especially for a pathology audience, because we are typically using hospital-based cohorts that are retrospectively collected. So I think this will be, hopefully over the next hour, we can start to think about ways in which, prospectively collected population-based cohorts can really answer many critical questions that are difficult to answer in our hospital-based cohorts. So very excited to hear her talk today, and take it away, Lorelei. Thank you.
Great, thank you both.
One
sure,
last
yeah.
housekeeping note I have on my ledger here. Just to remind everyone to submit your questions through the Q&A tab, and we will address them at the end of our presentation. Sorry, go ahead, Lorelei.
No, that's great. Thank you so much. And it's really such a pleasure to be here today. And it was really a great opportunity for me to sort of put together some of the thoughts and some of the studies that we've done really over the past 19 years. And I've been a collaborator with both Rohit and Tamara over those years. And I think those partnerships between pathology and epidemiology have really shed so much light into prostate cancer in a number of different ways. So I wanted to hear my disclosures, neither of which influenced the work I'm talking about today. So I thought I would actually take a step back and say, like, how does somebody who's trained as a cancer epidemiologist end up doing work in the space of tumor-based biomarker studies? And actually, so I actually trained not in public health, but in biology. Went out to Seattle after I graduated, worked in a basic science research lab, And I think there's a lot in my own career that I feel like serendipity has played a role both in my life, but in my science as well. I had a chance to move to Jackson Hole, Wyoming for about a year and a half before I started graduate school, and worked with the pathologist at the local hospital who actually covered half of Western, like all of Western Wyoming. And it was there that I really first got introduced as a histopathologist about both the complexities of a pathologist who covers every aspect of pathology, but also about rural health in America. I'd never heard of the field of epidemiology up until that point, when actually a friend of mine had just started at the Harvard School of Public Health, and she said, "You really should check out this field of epidemiology. I think it would mix those interests of yours both in biology, but also having a population level impact." So along the way, I did a master's of public health in Boston University. I did my doctoral training at the Harvard School of Public Health, and then went to the Karolinska Institute. And it was actually there that I became, this is a lot of sharing of information, but I think it's helpful to share. Like I was pregnant with my son. Ethan at the time wanted to come back to the United States, and there was an opportunity to come back to Harvard School of Public Health and lead a number of initiatives of tumor-based biomarkers in prostate cancer cohort studies. So today I'm going to be hoping to talk with all of you about the role that patho-epidemiology studies that are nested within prospective cohort studies can really help shed light on three different areas. The first is thinking about these studies and how they inform our understanding of the etiology of prostate cancer, specifically as it relates to molecular subtypes of prostate cancer. Secondly, how we can use both epidemiologic data as well as tumor biomarker data to help us understand mechanisms and establish causality with specific risk factors for prostate cancer. And then finally, as Tamara mentioned, thinking about how we can use our cohort studies to really credential and add additional value to those studies that are done in the hospital setting to credential prostate cancer prognostic markers. So I first wanted to give you an overview of a cohort, and actually Tamara is our lead pathologist on the study, the health professionals follow-up study in our prostate cancer studies. So this was a really interesting cohort. We're reaching our 40th year of this cohort going on. It started with 51,000 male health professionals. So these were dentists, optometrists, veterinarians, pharmacists, et cetera. And the idea was that these individuals, by focusing in on male health professionals, would probably be in the best position to talk about their own health, and also that they'd be likely to stay committed to doing a study. So we follow these individuals every 2 years with detailed questionnaires that ask a range of factors from traditional lifestyle factors, dietary patterns, medications. We also ask them to report about their health outcomes, including cancer diagnoses. And then we follow up those self-reports and get medical records and pathology reports on these individuals. We also have a whole range of different biomarkers, biospecimens and biomarkers that we've collected in the cohort. The first is we actually have one of the largest repositories of toenails, which can be very helpful for studying things like heavy metals, selenium, et cetera. In around 1993 to 1995, we collected a prospective blood specimen from about 18,000 of the participants. And then we followed up around 2004 and collected buccal cells from individuals for whom we did not have blood so that we could do DNA-based studies on them. We have a small but very deeply phenotyped cohort within the health professionals follow-up study where we've collected urine, blood, and stool specimens at multiple time points that have allowed us to validate a whole range of different factors. And then what we've done is to go back and retrieve the tumor tissue specimens, not only for prostate cancer patients, but actually for 10 different malignancies, to retrieve the clinically collected specimens, so either from biopsies or surgical specimens. And we now have those based as a tumor biorepository at the Harvard School of Public Health. So we have, of the almost 9,000 individuals who've been diagnosed with prostate cancer over the past 40 years, we have about the tumor tissue materials and slides for about half of them. And actually, just to add, what's really remarkable, again, I said that we've been following these men now for 40 years. We still have 16,000 individuals who are actively follow up. Our last completion rate of our survey was 90%. So we really have high participation and also really long-term follow-up and detailed exposures both before cancer diagnosis and after. So I wanted also, although I'm going to be talking mostly about the tumor-based biomarkers in the cohort, I wanted to give you a sense also about the other types of DNA, plasma, toenail, and other biomarkers we've collected over time, just so that I'll be talking a little bit about some data that we have linking germline based biomarkers together with the tissue-based biomarkers, but we really have a detailed set of markers collected on the cohort. So as I mentioned, we now have tumor tissue specimens, both prostate biopsy tissue from 1,767 of the individuals diagnosed with prostate cancer in this cohort, as well as radical prostatectomy tissue specimens for just over 2,500 of the participants. From the prostatectomy tissue, we've made tissue microarrays. So currently we have 21 tissue microarrays, and these include, on average, 3 cores of tumor tissue per person diagnosed with prostate cancer, as well as normal tissue as well. And we've done an array of immunohistochemical markers, some of which I'll present here during this talk. We've also done a detailed histologic evaluation of re-reviewing for Gleason score, aspects of inflammation, perineural invasion, and other histologic features, which is important because our participants are from all over the United States. Some are diagnosed with prostate cancer in community hospitals, some in large cancer centers. So to standardize the histologic aspects of the cohort, these have all undergone standardized histopathology review under the leadership of Dr. Lotan and her colleagues. So the first set of studies I'm going to be presenting are around how we can think about prostate cancer etiology through the lens of molecular subtypes of prostate cancer. So if you were to ask a cancer epidemiologist, what are the risk factors for overall prostate cancer risk? I would say these are probably the 5 factors that everybody would agree on. So older age, individuals of African ancestry. Actually, now in the United States, we're seeing an excess risk of prostate cancer also among Native American Indian populations. Having a positive family history of prostate cancer as well as breast cancer are strong risk factors for a man's future risk of prostate cancer. And we've now, through assessing both more common genetic polymorphisms as well as more rare genetic variants, particularly in DNA repair pathways, have credentialed a number of individual genetic risk loci, which we are associated with strongly with an increased risk of prostate cancer. And then interestingly, I think taller height is a risk factor for many cancers, including prostate cancer. One of the challenges with these risk factors, however, is one, they only explain a proportion of the cancer. Secondly, they're not modifiable. And what we do know about prostate cancer, as we do with other cancers, however, is there's a lot of heterogeneity clinically, in terms of risk of metastasis. In addition, molecularly now, we really understand the molecular heterogeneity of these cancers. So our thought is perhaps there are additional risk factors that can be credentialed for prostate cancer if we integrate molecular subtypes of the disease. And so one of the early characterizations of defining what the molecular subtypes of primary prostate cancer are, and this is going to be different than what you might see for metastatic prostate cancer, but for primary prostate cancer, this was data based on the Cancer Genome Atlas. There's essentially thought to be 7 specific subtypes. The most common of the subtypes involves gene fusion events that involve the members of the ETS family of transcriptional factors. The most common one is ERG, bound to TMPRSS2. There's also a little bit more rare gene fusions involving other ETS partners, including ETV1 and ETV4. And that encompasses about, say, 60 to 70% of primary prostate cancer have one of these gene fusions. The other common subtypes involve mutations in genes SPOP, FOXA1, and you can see IDH1, but that's in primary prostate cancer, it's quite rare. And then there's about a quarter of prostate tumors where we not sure exactly what the primary molecular subtype of that is. So a lot of the work that I've been doing really over the past, I would say 15 years, has involved etiologic studies looking at potential risk factors for prostate cancers based on presence of this TMPRSS2 gene fusion. And so about 50% of all prostate cancer tumors harbor the gene fusion. In the United States, given the incidence rate of prostate cancer, that translates to about 150,000 patients diagnosed each year with TMPRSS2 fusion positive prostate tumors. I think quite interestingly, however, the prevalence really varies based on ancestry of patients, with a lower prevalence of TMPRSS2 in Black patients specifically, which is interesting also because they are a group of people who are at greater risk of dying of prostate cancer. So it's a gene fusion. Both genes are located on chromosome 21, and it can involve either a deletion event or a translocation event to result in this gene fusion. TMPRSS2 is felt to be an early oncologic event. And what's also quite interesting is that these other non-ERG fusions that involve ETS partners seem to be in some ways transcriptionally, when you look at the transcriptome of their tumors, similar to TMPRSS2. So I'm highlighting here some work led by our colleague Konrad Stopsack, who did a gene expression profiling analysis, including the Health Professionals Follow-Up Study, Johns Hopkins-based cohort, as well as the Cancer Genome Atlas. And I would say that's one of the things I think that's really quite important with tumor biomarker studies, is an opportunity to validate findings across multiple settings. But one of the things that Conrad noticed in his analysis was pretty big differences between ERG positive prostate cancer cases, or TMPRSS2 positive compared to not, and also that the non-ERG fusions were similar to the ERG fusions. And two important pathways that popped up were fatty acid synthase metabolism and amino acid metabolism that were overexpressed in TMPRSS2. We've also done some work showing protein expression of insulin receptor and IGF-1 receptor, so other aspects of lipid metabolism seem to also be overexpressed. And that's also pretty interesting given the similarities of gene fusion events in Ewing sarcoma that involve another ETS partner called FLE1. And these tumors in Ewing sarcoma are also associated with upregulation of both insulin and IGF-1 receptor, and there are targeted therapies in that space. So we recently undertook a systematic review of all of the literature of studies that have looked at etiologic risk factors for prostate cancer based on the TMPRSS2 gene fusion. This was led by a PhD student in our group, Colleen McGrath. And this is a summary of the studies thus far. So although the gene fusion was first identified now, it's actually almost been 2 decades that it was first identified. There's not a huge body of literature that has looked at TMPRSS2 etiologically, despite how common it is. So these are the studies that we included in our systematic review. I first wanted to touch upon some of the results on genetic susceptibility to prostate cancer, and specifically how it relates to TMPRSS2. So prostate cancer turns out to be really interesting as the cancer with the highest estimate of heritability, meaning the highest or strongest contribution of inherited genetic factors to the overall variability and risk. And this is data from a study I did in the Nordic Twin study of cancer, which is a population-based registry of twins in the Nordic countries. It included over 200,000 Nordic twins. And what's interesting about twins to research on is that you have two different types of pairs of twins. You have those that share, in theory, all of their inherited genome. Those are the identical twins. And then you have familial twins or dizygotic twins who share on average about half of their inherited genome. So you can kind of compare the concordance of prostate cancer and twin pairs between these two different groups of twins to get at an estimate of heritability. So based on this twin study, about 58% of inherited of the variability in risk of prostate cancer is due to inherited genetic factors. And this is just to show here that prostate cancer really stands out, whether you look at twin-based studies or genome-wide association-based studies of heritability. Prostate cancer really stands out as the strongest germline component. So we have studied Tempur-Stewart in our health professionals follow-up study cohort, leveraging a validated immunohistochemical marker of ERG overexpression. So this correlates quite well with more fish-based approaches or other types of assays as a good marker of the Tempur-Stewart fusion. So this is in our cohort, about half of the tumors of prostate cancer patients in our cohort contain the Tempur-Stewart gene fusion, sort of similar characteristics with respect to age, Gleason score, pathology stage, PSA values. And I think this is seen in other studies as well. It's not particularly prognostic on its own, but etiologically and together with other factors, it may be prognostic. Also, what we see, interestingly, much more associated with PTEN loss, which again has been seen previously, and then also seems to have a higher prevalence of mutations in P53. So the first study I want to highlight is work now that was done on an earlier set of validated germline risk factors for prostate cancer. Again, these are more common single nucleotide polymorphisms, and this was a study where we had both germline data and tumor tissue data for about 500 prostate cancer cases. So we knew their ERG status, and then we had individuals who did not have prostate cancer. And out of these 39 individual SNPs, we saw 10 of them were either specifically associated with an increased risk of Tempur-Stewart ERG, specifically associated with a risk of ERG-negative prostate cancer, or their association with risk was actually in the opposite direction. So just by chance, we would have expected 2 of these SNPs to pop up. So this really seems to potentially indicate that the different germline factors may predict prostate cancer risk separately for the ERG positive and ERG negative prostate cancer. So I think that's something we hope to explore in larger studies. Another germline-based study we did was based on the role of the androgen receptor in the development of Tempur-Stewart ERG. So it seems experimentally that androgen receptor, or AR signaling, induces spatial proximity between the two genes given their location on chromosome 21. And together with radiation, it increases the probability of a fusion event occurring. There's a common repeat length polymorphism, a CAG repeat polymorphism in the androgen receptor, where the larger number of the CAG repeats in the germline, the inverse association with transcriptional activity of the androgen receptor. So you have a germline polymorphism that predicts transcriptional activity of the androgen receptor. So we looked again within the health professionals follow-up study where we had characterized the androgen receptor CAG repeat length and looked at how it was associated with the risk of Tempur-Stewart positive versus negative prostate cancer. And so these are odds ratios of prostate cancer. The blue line is the risk of prostate cancer that contains Tempur-Stewart, and the red line represents prostate cancers that lacked the Tempur-Stewart fusion. And again, the shorter the CAG repeat length, the greater the transcriptional activity of the androgen receptor. And based on what our hypothesis was, and in line with that, we saw no association of CAG repeat length in the germline and risk of prostate cancers that lack Tempur-Stewart, but we see a strong and positive increasing trend with shorter repeat length and ERG-positive prostate cancer. So again, in line with our hypothesis. We then followed up with a study. We have circulating hormone levels, sex hormone levels that were measured on average about 10 years before diagnosis of prostate cancer. And in a study, and this was fairly small numbers of individuals with prostate cancer and Temperature data and the circulating hormone levels. However, the study was led by Rebecca Graff. We looked at a whole range of different sex hormones. The one that really stood out, which again was quite interesting to us, was that higher levels of free testosterone specifically seemed only to be associated with an increased risk of ERG-positive prostate cancer. So essentially no association with ERG-negative disease. So again, aligning with this hypothesis, and it's interesting that TMPRSS2 is a gene that's highly regulated by androgens, and that androgens signaling can lead to, experimentally, the formation of the gene fusion event in cell lines. So it's quite interesting, this hypothesis around the germline CAG repeat and levels of free testosterone as well. Another and kind of very different study that we did was looking at certain factors, epidemiologic factors, that we know have an influence on a range of different metabolic pathways, sex hormone levels, et cetera. This particular set of data was looking at vigorous physical activity. So we had, again, we have a cohort of men, 50,000 of them, who have reported every 2 years on their levels of physical activity. These are quite active men, actually. So we're looking at equal groups of physical activity. So the quintile 5 is the higher level of physical activity. This is averaging about 30 minutes per day, 5 days a week of vigorous physical activity. So that means getting the heart rate up, compared to men probably who are a little bit more sedentary. And physical activity really has such broad effects on metabolic health, inflammatory markers, and other hormone levels. And what we saw, and again, this was in line with our hypothesis, was that although vigorous physical activity did not seem to have an effect on prostate cancer risk based on ERG-negative tumors, there was a strong inverse association such that the men who had higher levels of physical activity had a much lower risk of TMPRSS2 ERG-positive prostate cancer in the future. So again, very interesting, kind of provocative findings. So when, kind of in summary, and this is based in the meta-analysis, we tried to think, how do the direction of these associations with TMPRSS2 ERG tell us something about potential mechanisms by which these factors might be influencing the risk of TMPRSS2 ERG-positive prostate cancer. So, I'd already mentioned sex hormone signaling, androgen receptor signaling for free testosterone, AR-CAG repeat length. We also see taller height, as well as circulating levels of different lipids seem to be associated with specifically with ERG-positive prostate cancer, which may indicate a role of IGF-1 in insulin signaling, as well as altered metabolism. We also saw some really interesting findings around lycopene, which is an antioxidant, which is found in quite high levels in cooked tomato products. Interestingly, the prostate accumulates lycopene at high levels when men eat cooked tomato products. It's also in watermelon, other fruits and vegetables. So we're thinking potentially there may be a role of oxidative stress in lowering the risk of ERG-positive cancers. So just some of the takeaway on the etiologic studies, we really think that TMPRSS2 ERG represents a unique molecular subtype of prostate cancer with its own set of risk factors. And some of these risk factor associations shed light on some of the molecular mechanisms by which this fusion might be occurring. I think one of the strengths of doing this in patho-epidemiology studies is that we have, as I mentioned, diverse data elements, both before and after cancer diagnosis, as well as long-term follow-up and other types of biomarkers. And I think we hope some of these studies could shed light on opportunities for precision prevention and may guide development of randomized clinical trials. Are some of the future studies, and Konrad Stopczak and Tamara together are leading studies to integrate non-ERG-ETS fusions into these etiologic studies. So I'm going to go next to thinking about how patho-epidemiology studies can inform mechanisms of risk factors, and I think also may help potentially identify novel targets that could be for therapeutic intervention. So I'm going to use obesity as a theme for this. In obesity, although it's not associated with overall prostate cancer risk, there's quite good data now that excess body weight is a strong risk factor for development of advanced prostate cancer. And in cancer patients, obesity is generally associated with worse outcomes, worse response to androgen deprivation therapy, and higher cancer mortality. And data from a 2014 report from the World Institute of Cancer Research suggested the evidence was strong for its association with more advanced prostate cancer. These are data from Barbara Dickerman, who was at the time a student of mine, looking at a cohort of men from Iceland, looking specifically at abdominal obesity or different types of obesity to see whether there's a theory that the central obesity or visceral obesity is more metabolically active. It's actually interestingly more sensitive to things like diet and physical activity. But the question we wanted to ask was abdominal visceral adiposity a stronger risk factor than, say, body mass index or other measures of body fat? And in fact, that is what we saw. We saw about a twofold higher risk of advanced prostate cancer for men who had greater visceral adiposity. So how might obesity influence more aggressive forms of prostate cancer? So I think we wanted to bring in the Paget's seed and soil hypothesis that obesity may potentially have more systemic effects on immune system, metabolic health, et cetera, that may drive metastatic disease. But we really wanted to look at the local seeding effects. Does it influence tumor biology? And if so, is it setting up the tumor and priming the tumor to a more aggressive phenotype? So one of the first studies we did around this was asking the question of, in patients who already have prostate cancer, does obesity, are the ERG positive tumors, if they are associated with higher insulin signaling, fatty acid synthase, other metabolic factors, are they going to be more sensitive to the effects of obesity on survival? And so this was a study in the health professionals follow-up study, so patients only, so they already had prostate cancer. And this was looking at central obesity or waist circumference. And in fact, that's what we found was there was essentially no association between obesity and prostate cancer survival in those tumors where ERG was not present, but a strong and positive association where the fusion was present. So greater obesity, increased death from prostate cancer if they had TMPRSS2 ERG fusion in their prostates. And so then we asked a kind of a larger question, which was, can we see broader tumor-specific differences, or even are there differences in the microenvironment if we look at the transcriptome level? So we had previously done a transcriptomic analysis characterizing about the mRNA expression of about 20,000 genes in 425 prostate cancer patients within the health professionals follow-up study, as well as for half of them, we also had profiled the normal tissue as well. And then we asked the question of, are there genes or pathways or genes at the transcriptomic level that differed in individuals either who had a healthy weight or individuals who were considered very overweight based on body mass index? And this was led by Svetlana Tekacheva, who's at the Dana-Farber Cancer Institute, and Erica Ebot, who was a postdoctoral fellow with us at the time. And what was really quite interesting to see was when you looked not at the biological processes that were enriched in the prostate tumors of overweight men, many of them, in addition to aspects of metabolic health, were related to chromatin modifications or epigenetic related changes. And that was pretty interesting in that we were also part of a collaboration that was led by Miles Brown and David Labe, Miles Brown being at Dana-Farber, looking at a mouse model of diet-induced obesity. And when we looked at the transcriptomes of the tumors in the mice to see how similar they were to what we saw in the human prostate, we saw something very similar. So that was really interesting to see. And then we took the top 35 genes at the mRNA level that contributed to these chromatin remodeling signatures, and we asked the question, are these on their own, if we had a gene score in the tumor, are these on their own associated with lethal prostate cancer, either death from cancer or metastatic disease? And even if we adjusted for the stage or grade or age of the person, we see this strong and positive association with a fivefold excess risk of prostate cancer compared to those with the lowest expression of the chromatin score. And then what was also interesting, and what we hypothesized, that although part of the effect of obesity on development of lethal prostate cancer may be through expression of the altered chromatin, there's also an independent effect of obesity on lethal prostate cancer that we couldn't explain. So this was pretty interesting to us. And then we've done a couple of other studies of integrating obesity and tissue-based biomarkers. One was on looking at tumor angiogenesis, which may be one of the key pathways to help facilitate metastases by supplying tumor cells with direct access to the blood system through its vasculature. We characterized tumor angiogenesis in the health professionals follow-up study tumor cohort, and really found, particularly in the high Gleason grade tumors, that there was a strong differentiation in risk of death from prostate cancer based on the extent of tumor angiogenesis. So extent of tumor angiogenesis seemed to be a strong risk factor for death, even in these high-grade cancers. And so we asked the question of, what about changes in body weight over time? And we were able, and how does that relate to tumor angiogenesis? And what was interesting, what we were able to do was, because we have data on men not only at every 2 years in the cohort, we actually had asked them to tell us about body weight at age 5, age 10, age 20, so even before the study. And we came up with these 4 different patterns of weight trajectory over time. We have one pattern where the men were essentially pretty lean early on in life, but gained either moderate or in the orange to an extreme level of body weight. And then also we had individuals who stayed they were perhaps a little bit more moderate early on in life, but stayed pretty stable. And then we had a group that was initially quite heavy early in life and who stayed pretty heavy throughout life. So 4 basic trajectories of men over time. And what was really interesting to see was, compared to those men who were lean or early in life and whose weight, kept them pretty stable, so they weren't gaining a lot of weight over time. They were the reference group. In particular, the lean men who showed moderate increased weight over time, as well as those who were heavy all along, they saw substantially increased tumor angiogenesis, and in particular, really strong difference in excess risk in those with the high-grade tumors. So really interesting effects of body weight. And I'm going to just skip these 2 just for the interest of time. So I think in terms of thinking about these studies of patho-epidemiology studies of tumor mechanisms, I think obesity really highlights a really interesting, potentially modifiable risk factor for advanced prostate cancer. And I think we hope that some of the biomarker-based studies help also establish causality of the association, as well as may give some insights into other actionable biomarkers to prevent the progression of advanced prostate cancer. And it may highlight also some unique tumor features of individuals who may be more susceptible to the effects of obesity and therefore might benefit more from weight loss. So in the final few slides, I'm going to just highlight a few of our studies of tumor-based biomarkers in our patho-epidemiology studies that inform prognosis of prostate cancer. The first is sort of perhaps not as an exciting biomarker, but I think such an important clinical factor in predicting metastatic and fatal prostate cancer over time, is the Gleason score. And because of our cohorts, we thought it would be quite interesting to look at the long-term performance of Gleason score over 30 years of follow-up from diagnosis, given the long natural history of prostate cancer. And we also wanted to ask the question of, do you see Gleason score predicting metastatic and fatal disease similarly over time? Or do you see that it's more prognostic either earlier after diagnosis or later? And because these are cases diagnosed all over the United States, they're not only from say a high-risk population, they really represent the Gleason scores that you'd see kind of at a population-based level. So these are the cumulative incidence over 30 years of lethal prostate cancer, and you can see pretty big differences. The table sort of summarizes it too, looking at the risk difference. Basically, if you compare the lowest Gleason 6 tumors, the low risk of metastasis, compared to those at the highest risk after 25 years, you're seeing about a risk difference. Actually, we're actually comparing it to the 3+4. You're seeing about a absolute difference of 27% for lifetime risk of lethal prostate cancer. These are all men who were not metastatic at diagnosis, and even seeing big differences in lifetime risk of lethal cancer among the survivors who lived 10 years metastasis free. So seeing discrimination. And then these are the time varying areas under the curve, as well as the confidence intervals, looking at 5 years after diagnosis, 10 years after diagnosis, 15 years, 20 years, 25 years. And just a couple of take-home messages. One is the strongest risk prediction we saw across ages, and whether it's Gleason alone or with other clinical factors, was predicting lethal prostate cancer up to 10 years. However, we still see really good prognostic value of Gleason score as well as clinical factors across time, even 25 years in the future. Also, what was interesting too was there was a suggestion that maybe Gleason score was more prognostic in those individuals diagnosed in the less than 60 years or even 60 to 69 years compared to the older individuals. So the next project that I wanted to highlight was some work that we did together with Angelica Ammon, who was at MIT and just a tremendous molecular biologist who was interested in aneuploidy in cancer, and had looked at the data from the Cancer Genome Atlas and had seen, as others had seen, that compared to other cancers, which tend to have a lot of copy number gains and losses across the genome, prostate, as you can see, which is in where the arrow is, tends to have a lot less tumor aneuploidy. So she wondered whether at the population level, could tumor aneuploidy be a strong prognostic factor? So we looked at this, and this was again led by Conrad Stopsack within the Cancer Genome Atlas and the Health Professionals Follow-Up Study to ask the question, can we didn't have at the time actual DNA copy number of the tumors, but we had the transcriptomic data. So we said, can we predict from the transcriptome tumor aneuploidy, and how does that relate to cancer prognosis? And this table shows here, looking at for each of the chromosomal arms, the top figure looks at the prevalence of aneuploidy for that arm. So you can see the highest prevalence is for loss on the HP chromosome arm, and then the largest is gain of HQ. But you also see in 13q, 16q loss, as well as 18q. So about 1 in 5 prostate tumors, and again, these are individuals who did not have metastatic disease at diagnosis. About 1 in 5 showed really considerable extent of aneuploidy across 5 or more chromosome arms. And what the study found was that even adjusting for other prognostic factors such as Gleason score and clinical factors, that those individuals who had higher levels of aneuploidy, so those 1 in 5 tumors with 5 or more predicted chromosome arms, had about a 5-fold greater risk of lethal prostate cancer. But even those with 1 or 2 or 3 or 4 had about a 2-fold greater risk of lethal prostate cancer. So pretty interesting findings. So the last study that I wanted to highlight, and I think it touches upon the importance of validation across multiple cohorts, is a study of work that Conrad led together with Tamara, looking at a validated immunohistaining for p53 mutations on our TMA cohorts in the health professionals follow-up study. So the work of using immunohistochemistry to validate p53 mutations was done previously, and then that biomarker was integrated into our tumor tissues. So in primary prostate cancer, p53 nuclear accumulation is not common. In our cohorts, it was prevalent in about 4% of tumors. However, these tumors are really aggressive. So correlated with greater loss of P10, a lot more Ki67 proliferation. This figure B shows a greater extent of tumor aneuploidy associated with p53 positive tumors. And then the other factor that was quite important, oh, sorry, it didn't come here, but was that p53, presence of p53 mutations was strongly prognostic, again, outside of Gleason score, tumor stage, et cetera, but also that at the univariate level, that the p53 tumors, not only were they more likely to die, they were more likely to die about 4.6 years sooner than those without the p53 mutation. So again, I think what's interesting about doing a study like this in our cohorts is, again, these are not highly selected, more aggressive patients. These are kind of the population level of what you might expect to see with p53 alterations, but showing that they're strongly prognostic. And we have some unpublished data. It was a little too early to show today that it looks like p53 remains strongly prognostic over 25 years. So again, thinking about that time varying area under the curve, it seems that p53 remains strongly prognostic over decades of follow-up. So just in summary, I think I just wanted to highlight how important it is to work with pathology colleagues on studies like this who have validated biomarkers genomically and clinically to inform prostate cancer prognosis. And as I mentioned already, these studies, when you nest them within cancer epidemiology cohorts such as ours, they really don't have the same selection forces that you'd have in a clinically based study. Also, we have the advantage that we have just long-term decades of follow-up. And the final thing that I wanted to highlight is that when you look at indolent cancers that even after following for 20 years, there's no development of metastasis, there's a lot of common genomic features of the tumors. The Gleason score is similar. They tend to not have P10 loss, don't have P53 loss. They lack aneuploidy. But when you look at the tumors that have a high risk of metastasis, there's a lot of different mechanisms that might be upregulated. So it might be more aneuploidy, P10 loss. It might be high Gleason score, it might be angiogenesis. And so this idea that prostate cancer progression of lethal prostate cancer can occur in a variety of ways. And it's sort of similar to Tolstoy's statement in Anna Karenina that all happy families are alike, but each unhappy family is unhappy in its own specific way. So I'd like to thank so many people for this work that I presented today. It's been an incredible collaboration with really people all over the globe. And I'm just excited to hear your questions and look forward to talk more. And maybe I'll stop sharing my slides then as well.
Great talk, Lorelei. That was fantastic. So we have a couple questions in the Q&A, which I'll start off with. The first question, I guess, is about 5-alpha reductase inhibitor use, like finasteride. Do you think TMPRSS2 ERG fusion may be arising because of unconverted testosterone, or do we know anything about ERG in the PCPT trial? Or yeah.
That is a really great question to which I don't know the answer, but I'd love to follow up on that. One is, when I was, one of the reasons I wanted to highlight some of our studies is that if people have hypotheses they'd like to explore within our cohorts, we actually have data on 5-alpha reductase inhibitor use across time. So it's a question we could actually ask. I don't know if anybody's asked that question. Biologically, to me, it makes a lot of sense. But it's something we could actually test within our cohort studies. So if the individual would like to reach out to me about collaborations and with us tomorrow, I think that would be fantastic.
Yeah, really interesting question. Second question, circulating lipids seem to increase risk and adiposity reduces risk. Is this contradictory?
This is a great question. So I think what we're starting to understand about adiposity is really it's important about the timing of adiposity. So what we know is that obesity, and it's actually analogous to breast cancer as well, where obesity early in life is actually associated with a lower incidence of breast cancer later in life, whereas adult obesity, particularly after menopause, is associated with an increased risk of breast cancer. And we think it may be similar in prostate cancer. You know, the prostate really undergoes extensive growth and maturation during puberty. So if a boy might have excess body weight during that time, levels of sex hormones are going to be lower, such as levels of testosterone. Whereas later in life, and so maybe what's important around puberty is the fact that sex hormone levels are low. Whereas later in life, where we are starting to see a positive association of later life obesity and advanced prostate cancer, it may be more important that the metabolic consequences of obesity. So we're really thinking that it's a complex association, and trying to understand that complexity over time is something that's really important. But I think that may be what's going on.
Good question. Any morphologic features correlating with aneuploidy? And we had another similar question where the aneuploidy changes you saw different across different Gleason scores from low to high.
Yeah.
Out within different clones that had different Gleason scores within one patient.
Right. So that's actually the second part of the question is not a question that we could address the way we did our study, because we were looking at sort of the bulk tumor and doing transcriptomes on that. I think it would be fascinating to do some sort of in situ assessment where you could really look at the correlation between directly of different histologic features. Also to examine, do we see anything in the adjacent stroma? It was interesting when we looked at whether any presence of aneuploidy in the normal tissue adjacent to cancer. We don't see much going on, so it really does seem to be a tumor-specific effect. But there were tumor aneuploidy was strongly related to the Gleason score, not surprisingly. But we even did see in the Gleason 7 tumors, if you look at that subgroup, extent of aneuploidy it was prognostic and could really distinguish risk in that more narrow Gleason score. We haven't looked specifically, and it would be interesting, again, this is if somebody's interested in doing this study, we have all of these other histologic features. We could look at those associations with extent of aneuploidy. So if somebody were interested, and we could also, I know within the Cancer Genome Atlas data set, I believe there's images of the prostate tissue there as well. So we could look in that cohort as well, because the aneuploidy has been characterized using DNA copy number specifically.
Yeah, yeah. And another individual suggested any correlation with criviform or intraductal as well.
Yeah, yeah, that would be fascinating to take a look at. Yeah.
Let's see. Do you think immunohistochemical detection of testosterone in prostatic acinar cells in TERP can predict genesis of prostatic adenocarcinoma?
Oh, that's a
Anything
really interesting
could be done
question.
in situ? Yeah.
Right, right. I don't know. I guess maybe a question to the two of you. Can you detect, how would you detect, are there validated antibodies to detect testosterone specifically in prostate tissue? I know we could look at androgen receptors specifically, but I'm not really sure about measuring the ligand specifically. I'm not sure.
Yeah, maybe with in situ hybridization or something. Yeah. It tends to be difficult to detect secreted things and yeah. But I guess in situ might be one way.
Or like a metabolic product.
Yeah.
Yeah.
I think the other thing that you could be really interesting, and I know there are some groups that have looked at metabolomics within tissue-based metabolomics, and even surprisingly, Massimo Loda did some interesting work where he had paired fresh frozen and formalin-fixed tissue, although a lot of the lipid metabolites sort of got washed away with the fixation process, it seemed that there were some metabolites that remained even in the FFP tissues. So there could be some interesting ways to look at other biomarkers that way as well in the tissue itself. But you might need to work with fresh tissue for that, for some of them.
One question I had is that you mentioned the toenails at the beginning. Are you guys thinking about doing more exposome work, or where does that stand in prostate?
Yeah, no, I think it's such a great question. So we have a pilot study going on right now to look at whether we can measure some of these forever chemicals, like the PFAS exposures in the toenails themselves. So we have selected some of the individuals who have both toenails and where we've already measured blood levels of PFAS. So we want to be able to look at those correlations. The other interesting resource that we have was when the bloods were collected on the men, they because they were health professionals, they collected them themselves in some capacity, and then they sent the blood tubes back to us on ice packs that were from tap water they collected in their home. So we actually have all those tap waters stored. So we want to look at, for example, at PFAS measures. So that's one area that we're really interested in. We have a colleague who's done some interesting pilot work looking at heavy metals in the toenails. And I think heavy metals are super interesting. And maybe I think an interesting question is, why do all these things accumulate in the prostate? So cadmium, chromium, when men are exposed to high levels, for some reason, the prostate tissue is one of the organs that where there's really high levels of accumulation. I don't know why, but they do accumulate. So heavy metals could have that seed effect on the prostate itself. But then also these heavy metals can accumulate in the bone environment. So then you could think of it more of the soil hypothesis, that it creates an environment for, and it displaces calcium. So it could create a niche for lesions that accumulate in the bone metastasis to thrive. But I don't know why they do, but those are some other interesting areas in the exposome. I don't know if you have other hypotheses. And again, like if there are people out there who'd be interested in exploring a hypothesis, we'd love to work with people.
In the last couple of minutes, perhaps, I think one question I had was, I think very interestingly, you had mentioned that how androgen receptor signaling can induce TMPRSS to ERG proximity. And I think, if I remember, Dr. Ram Mani from Dr. Arul Chinayan's laboratory had similar, I think, findings that inflammation can actually induce also TMPRSS to an ERG proximity.
Yeah.
And I was looking at all of your slides, which is like fantastic work. I was just wondering, with the integral role of obesity now also associated with inflammation, like what do you think, how does inflammation, including prostatitis or prostatic atrophy or Angelo DiMarzo's work on PR lesions, prostatic inflammatory atrophy, what do you think, how inflammation might play out in this scenario?
You know what's so interesting? So we saw that paper, which was fantastic. And so we actually looked at just the correlation between the prevalence of these atrophic lesions, chronic inflammation, acute inflammation. And we actually see in our cohort kind of the opposite association, which is at least those histologic features of inflammation are lower in the ERG positive prostate cancers compared to the ERG negative cancers. But inflammation, there's so many, right? There's so many aspects of inflammation that we didn't look at. And so one question, I know one of the projects we're working on with Tamara now is to characterize immune cells. And I think that will be an interesting hypothesis, but there may be just other aspects of a more active inflammatory process that we're not capturing with those histologic features that might then tell us something.
Right.
And then one question we haven't looked at, which I think would be interesting, is what about systemic levels of inflammation like C-reactive protein or IL-6 or other inflammatory markers? Do those predict a higher risk of ERG positive versus ERG negative? When we haven't looked at that, and to my knowledge, I don't know if anyone's looked at that in in humans. So I think it's a super interesting hypothesis.
Okay. And perhaps, with Tamara's permission, last question. I think P53 mutant also has a null phenotype, so I think I was just curious. Actually, one of the I think our audience members also asked the same question. Perhaps that has been integrated into your studies. Is that correct?
Maybe I'll pass it to
Yeah.
Tamari since she's
Good question.
do all the validation of this work.
Yeah. So all we can detect, all we've been able to validate are detecting the missense mutations that stabilize the protein. At least in our hands, in the prostate itself, the endogenous levels of P53 are so low by immunohistochemical detection that it's hard to see
It's
loss.
hard. Okay.
So the
Got it.
truncation alterations we've not been able to reliably
Yeah.
detect with any clone that we've tried.
Got it.
So we are missing about a third of them. So that's a good point.
Got it.
We really only pick up the missense mutations, and that's probably why the frequency was especially low in this cohort. It's also a
Got it.
low grade cohort, but
Yeah.
yeah,
Right.
great
Thank you.
questions.
Thank you for sharing.
Yeah.
So I guess that was our last question, Tamara. Is that, can I close off the session with your
Yes.
permission? If that's okay.
Yeah.
Okay, sounds good. And again, before we close, I want to express my sincere gratitude and appreciation to our speaker, Dr. Mucci, for her time, expertise, amazing presentation, and thoughtful contributions to today's discussion. We had so many fascinating questions, and it seems like we might even have some projects in inception or evolution stage with the audience members, which is I think is fantastic. That is the whole point we are here today. I'm also very thankful to our session co-moderator, Dr. Tamara Lota, and I think who played such a big role in so many of the papers also, as shown by Dr. Mucci. And finally, thanks to all of you who joined us today, and we look forward to continuing these important conversations in our future GUPS events. Thank you and goodbye.
Thank you so much.
Thanks again, Lorelei. Bye-bye.
Thanks.